Okay, the next lab activity we have here, the acid-fast staining. So the acid-fast staining basically for identification of our acid-fast bacilli. So acid-fast bacilli include the group of your mycobacteria, nocardia, and we have also here the cryptosporidium. And they are identified here by a very special na cell wall structure made up of a thick mycolic acid. Theoretically, they are gram-positive pag na-perform tayo ng gram staining. Because of their thick mycolic acid, they try to resist here also decolorization pag nag-add tayo ng pure acetone if you perform here your acid-fast, or if you perform here your gram staining. But the best way for them to identify is through your acid-fast staining, not your gram staining. Okay, so basically they have here the properties that they have the thick mycolic acid where they try to resist here the decolorization if you're adding your acid alcohol when you're performing your acid fast staining. Pag sinabing they resist decolorization by acid alcohol, mean to say they tend not to be decolorized by an acid alcohol upon addition of your decolorizer when you're doing your acid fast staining reaction. Hence, you call them as your acid. You call that one as your acid fastness. So by sinabing acid fastness, they tend to resist decolorization by an acid alcohol. Mean to say hindi sila natatablan o hindi sila naapektuhan if you add here your acid alcohol. Okay, we have here the two methods here for the acid fast staining. One, we have your Zell-Nielsen, which is the hot method na gumagamit ng heat or the heating process. On the other hand, we have also your Kinyon. So this is a cold method. Wala itong heating process. So they are differentiated from one another by first, we have here the primer stain. The primer stain is carbol-fustin. Carbol-fustin is color red-orange. So the carbol-fustin of your Zell-Nielsen is made up of your basic fustin, okay, in 95% ethanol. Plus, we are also adding phenol para mag-penetrate itong ating primer stain para siya pumasok sa mga bacterial cell natin. Your Quinones, on the other hand, also have here the primer stain in the form of carbol-fustin. So it's also a basic fustin, only that nag-add ka ng wetting agent. So to enhance here the staining process. So wetting agent in the form of your tergitol, twin 80, 20, okay, so plus or phenol as well here to facilitate also here the taking up of your bacterial cell by the primary stain. So nag-add ka nito because walang heating dito. Walang heating dito. Heating process, aside from addition of your phenol, help here your primary stain, your carbol-fustin to be taken up by the cells natin para pumasok ang ating carbol-fustin. So since dito nag-heat ka, it would facilitate the entry of your primer stain plus your phenol. Dito kasi wala kang heating process, so kaya mag-add ka nito. That would enhance here the taking up of your stain, the primer stain of your bacterial cell. The colorizer here in our acid-fast bacilli or acid-fast staining is in the form of the acid alcohol. Acid alcohol because, okay, here in the Zell-Nielsen, this one is your pure or concentrated na acid, hydrochloric acid, in 95% ethanol, kaya acid alcohol. Acid plus or alcohol. Here naman sa ating Quinyon, so this one have here acid in the form of 1% hydrochloric acid lang in 70% na ethanol. Okay, now we have here the counter stain. So we have here your methylene blue. So blue ang color ng ating counter stain. Okay, here in your Zell-Nielsen, it's alkaline methylene blue. So methylene blue in 95% ethanol. It's alkaline because meron kang KOH, potassium hydroxide, 0.1% na potassium hydroxide. So providing here the alkalinity. Dito naman sa ating Quinyon, so it make use of the Loeffler's methylene blue. Okay, we have here the procedure for your Zell-Nielsen, which is your hot method. So procedure for that, so our specimen is in the form of your sputum. Okay, and then try to make a smear for the sputum. Okay, so medyo matrabaho ang preparation ng smear because again, remember the sputum is a viscous na specimen. So very thick ang ating sputum. So for that, you need to prepare that one in moderately thick lang na layers. So pag maglagay ka ng sputum, yung sputum natin parang sticky. So kailangan mo, so all you need to do is to spread, spread, spread. So with your applicator stick, ganun mo siya para ma-spread thoroughly ang ating specimen. So medyo pa isa-isa. So isa-isa mo ganun siya. Okay, until na mag-spread siya thinly in your film. Then after that one, dry. Okay, and then try to heat fix. Heat fixation again is being done here by passing your smear under the flame. Again, do not over flame that one. And after that one, you cover your smear here with a filter paper. Okay, make sure here that the filter paper has fully covered the entire smear. Then after that one, you flood. So pag may filter paper na, mag-flood tayo ng ating primer stain, your carbon-fusin. Okay, and again, you are adding here the filter paper to prevent the contamination of your smear by the stain precipitate. Para hindi magkaroon ng artifact formation. Because pag may more precipitate or artifacts here, mahirap mag-interpret at might have here some other reactions na maging erroneous ang ating interpretation. Okay, and after that one, you're going to heat. Okay, so pag may primer stain na siya dito, you try to heat. Okay, so heat mo over the flame. So make sure na mag-heat ka lang until na magkaroon siya ng steam. Okay, do not boil. And make sure na hindi siya nag-dry. Okay, pag nag-dry ang smear natin during the heating process, you need to replenish again here with your carbon-fusin. So, mean to say mag-add ka ulit ng carbon-fusin. Okay, allow to stand for 5 minutes. Then you remove the filter paper. Wash off with the tap water your slide. Then after that one, you try to add your decolorizer as alcohol. So just same way, pag nag-decolorize tayo ng ating acid, ng ating gram staining, so ganun pa din. Ganun mo lang siya, tapos, okay, tilt mo siya, then dito ka magde-drop para mag-flow dito sa ating smear. And make sure here that, you make sure that entire, okay, na carbon-fusin here na stain has been colorless or being washed off. Okay, so add your acid decolorizer until all the stains, carbon-fusin here na stains has been removed. Okay, then after that one, you wash again with the water ulit. Okay, then finally, you flood with the counter stain, which is your methylene blue. Okay, making sure that the entire smear is being covered by your stain. Okay, and then try to flood that one for 20 to 30 seconds. Usually, para mabilis na mag-flood ang ating stain, pag nag-add ka ng stain dito, like methylene blue, para mag-spread siya, gamit ka ng aspirator. So, ikanan mo siya para magkaroon ng hangin para mag-spread ang ating stain. Okay, then after that one, wash mo ulit siya. After application of your counter stain, try to wash off with tap water again. Okay, try to examine here under the OIO for identification of your acid-fast bacilli. Acid-fast bacilli would appear to be red in color, whereas non-acid-fast bacilli appears to be blue na color. Paano naging red ang ating acid-fast bacilli? Okay, remember here that ang ating, okay, so pag nag-add tayo ng primer stain, lahat ng cell mo or bacteria, regardless if it's one acid-fast or non-acid-fast, it will going to take up your primer stain. Since your primer stain is red in color, okay, so magiging red ang ating bacterial cell, regardless if it's acid-fast or non-acid-fast. Then, you try to add decolorizer. Okay, remember, pag acid-fast, it try to resist the decolorization. I mean to say, you could not remove the primer stain pag acid-fast bacilli. Pag non-acid-fast bacilli, they will be decolorized by your acid alcohol. Maging colorless siya. And it would allow your primer stain to escape. Pero pag non-acid-fast, because they have a thick mycolic acid, they would try to resist here the decolorization. So, walang effect ang ating decolorizer. And therefore, still, nandun pa rin si primer stain na carbon-fusin natin. So, red pa rin color niya. Then, if you try to add your contour stain, pag acid-fast bacilli, since andun pa sa primer stain natin, sa acid-fast bacilli natin, pag nag-add ka ng counter stain, hindi magpipenetrate itong counter stain. And therefore, retain niya ang primer stain color that gives red na color for acid-fast. In the case of your non-acid-fast, since after decolorization, okay, they try to remove here the primer stain. So, pag nag-add ka ng counter stain, they will going to take up here the counter stain, which is your methylene blue. So, magiging blue ang color nila.